I would like to say that it's a pleasure to be here in this virtual session to present our previous work. And today I will hold a communication about recent advances on biological particulate matter sampling techniques and characterization, but with a focus on fungal exposure assessment through indirect methods. This is my outline for today. Some background about waste sorting challenges regarding exposure assessment, the sampling methods and assays apply, and ending with the main findings. So, So waste sorting occupational environment is a challenging environment since the main source of microbial contamination is the waste to be sorted. And there is a scientific evidence that associates occupational exposure to bioaerosols, bacteria and fungi, in the waste industry to health outcomes. Among fungi, Aspergillus is the most prevalent genera on waste sorting, incineration, and also composting. As in other occupational environments that are prone in dust, we should consider, besides the fungal presence that was already assessed by culture-based methods and molecular tools, the occupational exposure to mycotoxins. In fact, in a previous study performed in this industry, was found exposure to aflatoxin B1 among workers. The suspicion of mycotoxins exposure was due to the Aspergillus contribution among the fungal contamination, as you can see in the figure with the red columns, meaning the Aspergillus contribution, and the gray area, very small, the other fungi. The azole resistance is also an occupational and public health menace in some specific workplaces. Climate change lead to an increase of fungi and mycotoxins contamination, causing the intensive use of azoles in crops to avoid toxigenic species, leading to resistant species that can multiply and survive. This is the case of Aspergillus fumigatus as an example, causing infections that are hard to treat and life-threatening. So in what concerns the sampling methods multi-approach, the waste sorting was one of the first studies performed by us that were used active and passive sampling methods. Two air sampling devices were used to overcome each limitations, and the device with impinge method was only used for targeting the harmful fungal species by molecular tools. At that time, 2014, we were only using surface swabs as passive sampling methods, and the results, either in air and in surfaces, follow the same trend, with the highest prevalence being from Aspergillus genera, and more specifically from Fumigatus section. In 2017, we did the first attempt in assessing fungal contamination in the filters from air conditioning belonging to forklifters that operate in the waste pile. It was interesting to observe that also followed the same trend regarding Aspergillus prevalence, and it was possible also to identify different results in counts and species when using different media, as can be observed in the table. Last year, we applied high-throughput sequencing in the same filters extracts, and it was possible to confirm the need of an accurate exposure assessment in the waste sorting industry. We also conclude about the wide spectrum of microbial contamination in this occupational environment, demonstrating that adequate monitoring of bioaerosol exposure is critical to evaluate and minimize health risks. Different occupational environments were assessed starting in 2018. We assessed different healthcare facilities, primary healthcare centers and central hospitals from Portugal. In this case, we combine air sampling with different passive sampling methods, such as surface swabs, electrostatic dust cloths, settle dust, and filters from heating, ventilation, and air conditioning systems. Both papers show the inefficacy of Portuguese legislation regarding indoor air quality, since we can comply with legislation if we perform only air sampling and count colonies. But if we decide to apply other sampling methods or even identify fungal species in the cases where the counts were complying, we can observe toxigenic species and some with clinical relevance. Additionally, in the primary healthcare centers were detected mycotoxins in air by Impingement method and also in settled dust. In a different study held also in a central hospital driven by workers' complaints, we used swabs from ventilation grids and also electrostatic dust cloths to avoid disturbance from that specific environment, and was possible a more complete characterization of the bioburden. We conclude that use of culture-based methods and molecular tools used in parallel should be the tendency to follow, and also about the need to analyze the azole-resistant profile for fungal species that might represent some health risk. An intervention including the use of UVC emitting device in the clinical pathology service where the study was held, and an increased maintenance and cleaning of the HVAC systems was recommended to promote the reduction of microbial contamination. Another setting assessed by us was the bakeries, and the study in Portugal was driven by the Portuguese culture regarding the bread. It is an essential food commodity in every Portuguese meal and employs an enlarged workforce. This study was granted by the Portuguese Authority for Working Conditions. We performed also a study in Ireland with similar sampling methods, and toxigenic fungal species were observed and detected by qPCR, and species with clinical relevance were observed on the azole resistance screening. The results also point to the possible exposure to mycotoxins, with 4 the probable contamination source, since different mycotoxins were detected in the raw materials. And both studies show the benefit of using a multi-approach regarding sampling methods, since different counts and species were observed in each sampling method. Last year, we decided to use protection device as sampling methods in the waste sorting. We started with filtering respiratory protection devices, and both interior layer and isolation valves were analyzed, applying culture-based methods and targeting for harmful fungal species and mycotoxins detection. These devices present high levels of bioburden, and were observed several Aspergillus sections with reduced susceptibility to the tested azoles and toxigenic fungal strains also. We also observed that workers with more waste contact show increased bioburden on these devices, and conclude about the adequacy to use them as passive sampling methods to assess occupational exposure. Regarding the interior of mechanical protection gloves, gram-negative bacteria were found in all. Aspergillus section with toxigenic and azole-resistant potential were observed, and mycotoxins were detected, and we should ponder about the exposure through ingestion route by hand-mouth in this case. We also analyzed the cytotoxic potential of these devices that could affect hepatic cells more than renal cells. The conclusions was that workers can have health effects due to the contamination found on mechanic protection gloves, and that these protection devices are adequate to be used as passive sampling method to estimate workers' exposure. Regarding the assays applied, in the previous slides, I also mentioned the use of culture methods applying more than one culture media, and molecular tools for targeting harmful fungal species. We are now using an algorithm in the lab work that allows us to obtain the microbial load and contamination, the detection of harmful fungal species by qPCR, the mycotoxins detection, and also to obtain the azo resistance profile with focus on the mutations detection by a deeper analysis of the aspergillus section fumigati isolates. In fact, and driven by the possibility that in some specific settings the azo pressure is higher, we apply the algorithm and we found in sawmills in full shift inhalable dust samples and in air samples from a dairy, and also in filtered respiratory protection devices from waste sorting, aspergillus fumigators harboring the specific mutation which is described worldwide as related to the use of azo fungicides in agriculture. These results support the theory that azo pressure is present in the occupational environment and in the environment in which fungicides are used, while there is a growing concern that the exposure to aspergillus, that the exposure of aspergillus to azo fungicides in the environment can cause cross-resistance to medical triazoles. Recently this year, we did a study including all the results from this specific section, the section aspergillus section fumigati, from the assessments that we have been performed since 2012. And besides giving guidance for an accurate exposure assessment, we also conclude about the need to perform the assessment by using active and passive sampling methods, to apply culture-based methods and molecular tools in parallel, and to not neglect the assessment of the microbiota azo resistance profile and the mycotoxins exposure assessment. So just to wrap up everything that I was talking about in the last minutes, we shouldn't use the air sampling as a standalone method. So depending on the occupational environments, we can combine and we should combine different environmental matrix. For instance, litter in the poultry, feed in the swine, mops in specific clinical environments. And we should adopt the passive methods to the possible contamination sources present in the setting that we are assessing. Also, we shouldn't use only one culture media. In our case, the use of TG18 in addition to mulch-strat agar allows for a broader characterization of contamination of the aspergillus genus in the workplaces. You can see in both figures that different media can give us different results, and they both should be considered for the exposure assessment. Also, we shouldn't use only culture-based methods. As you can see in these results from the mechanic protection gloves, in all the samples, the molecular detection allows to obtain a higher number of samples with the target aspergillus section, with the exception of Flavis section, since we were targeting only for the toxigenic strain that produce aflatoxin B1. Also, we shouldn't neglect the azo resistance screening in the workplaces, since our studies corroborate the azo pressure in some specific occupational environments, as in waste sorting industry, sawmills, and also dairies. And the last but not the least, we shouldn't disregard occupational exposure to mycotoxins. Mycotoxins should be recognized as a real occupational risk factors, being critical the need to accurately characterize mycotoxin exposure, which mycotoxins at which concentration and for which duration, and to understand which factors can influence that exposure. In some specific cases, like in bakeries, the assessment of raw materials and settled dust should be considered as passive sampling methods. So thank you for your attention. I will be happy to answer to some questions.
Thank you very much, Carla. Already a few pointers for follow-up questions, and that's great. Corinne, you got your hand raised.
I'll just give her a clap.
All right.
Good talk.
I think, yeah, you are raising quite a few already question on, so what we're going to have after the breakout session is already a great turn of people. We're 56, so we will break out maybe 5 or 6 sessions to discuss a bit further, because I'm sure you have been already raising some pointer on where we should and where we shouldn't, and maybe opportunity to exchange ideas and needs. So that was great talk for the setup. What I suggest is perhaps we might move to Rob now, where Rob is going to give us his views on within the regulatory context, but also the science on how it helped him and his colleagues around framing regulation, or trying to frame regulation and what we need and what sort of tool, but also how he's looking for working with the different stakeholder and academic to find the supporting evidence he needs to regulate properly and ensuring we are effectively providing the right advice. So Rob, over to you. I think you are more on going to share with us and providing us some open floor questions rather than presentation, which is nice also to do differently. And then after Rob's speech, you will get split in breakout session. But let's give the floor to Rob. You're muted, Rob. Rob, you're still mute.
Am I okay now? Sorry,
now
my
you
screen has gone unresponsive. That's great.
All right.
Okay. Well, thanks, Fred, for this opportunity, really, to set out our wish list, which all you very clever people are going to answer in the next couple of hours. Now, realistically, we've been working on bioaerosols for a long time now. I should say, first of all, that the agency to which I belong, the Environment Agency, is part of the UK Department of Environment, Food and Rural Affairs, DEFRA. So we're one executive part of government, and there are equivalents in the other home countries in Scotland, Wales, Northern Ireland. But we're all working together really to ensure that human health and animal health and plant health are not impacted by bioaerosols, particularly from the processes that we regulate, which primarily are agriculture and bio-waste in this context. So we do a lot on waste regulation. We do a fair bit on regulating large chicken and pig intensive agriculture, and we're now bringing cattle, various other aspects of agriculture into our regulatory sort of reach. And what we have to do in a nutshell is to make sure that the processes we regulate are not emitting more bioaerosol than is good for us. We have to make sure that exposure to people from these processes remains at an acceptable level. And that's quite a moot point in itself. What is an acceptable level? So the first challenge we faced when we really started focusing on this work is to say, really, what are the health effects of bioaerosol? Is there any evidence that bioaerosols are causing harm to human health? So a lot of the earlier work was literature searching really on impacts of bioaerosols. And as most of you will know, it comes primarily from occupational situations. We know that people working in various industries are prone to respiratory disease that can be associated to bioaerosols. But of course, these are all at very high levels. And what we didn't know was how low can you go? How low do you have to get in your bioaerosol concentration before you get to a level where you're not seeing adverse impacts, particularly out in amongst real people rather than workers, people who might be vulnerable in some way, who might be immune suppressed or particularly sensitive. So part of what we need to do is identify what are safe levels for various organisms. Part of it is to determine what within the bioaerosol mix is causing those adverse effects. And it's only through knowing those things that we can then set regulations, we can set targets for how much bioaerosol can be emitted, etc. So let me just go through then what we do. At the moment, as I say, we actively regulate pig and chicken farms and bio-waste sites, composting sites. We've done an awful lot of measurements for conventional culturable organisms and found safe distances, if you like, where the concentration has dropped down to something approaching the background level. And this is important too, incidentally. We need to understand that background in order that we can determine what perturbation our process is causing to that background. You know, not all Aspergillus exposure is coming from composting sites. You can get a big dose just from walking through a leafy forest, for example. So it's trying to work out proportionally what we're contributing to the bioaerosol burden. And quite a few people on the call have worked with us. I noticed Tony Gladding, you're on the call. All of the Cranfield guys have worked incredibly hard to take measurements which are repeatable, reproducible downwind of these various sites. And that's given the confidence for us to set target levels that will not be exceeded at sensitive receptors at the nearest place where people might be exposed to organisms. And that's well and good. We can regulate for that. We can say 4 times a year you will have someone come in, follow a protocol that we've defined, putting your samplers out at the same distance as the sensitive receptor and looking for whether you're exceeding guidance levels of Aspergillus as a sort of a marker for fungi, thermophilic bacteria and total bacteria. And that's the best we can do with the technologies available to us. We can take these snapshots, trying to make sure that they're at a time when most bioaerosol is being kicked off, and so they are representative. That's the situation at the moment then. What we're lacking is quite a shopping list. It's the understanding of that background that people are being exposed to. What's the context against which people are being exposed to bioaerosols? And to that end, we have this interest in working to better understand the atmospheric microbiome. How does it vary from season to season? How does it vary geographically? So what is it that we're contributing to in our process? When we're seeing illnesses caused by bioaerosol, is there anything we can do, or is it something that's endemic in the environment? There is this question of if there are lots of fungi, lots of bacteria coming off a site, but does it matter? We work closely with our public health colleagues in the Department of Health to try and work out what is, if you're not taking this completely precautionary approach that you will not exceed something approximate to background, how high can you go with exposure above that before you start to cause harm to people? So what we need for that is a lot more speciation of the bioaerosol that's coming off these sites. We've taken a sort of very tentative first stab at this. We've done some genomic work on bioaerosols captured downwind of pig and chicken farms, with the huge caveat that those samples weren't taken to conserve the DNA. We extracted DNA from them, but that was almost a spin-off, and of course it wasn't very representative because it wasn't done specifically for that purpose. But it was enough to encourage us that these methods can give us more speciation, and we can then go to the public health experts and say, right, look down this list. Is there anything that starts ringing alarm bells in particular? Which particular organisms do we really need to focus on? And that in turn gives us this option then of going to, rather than culturable methods, going to molecular methods such as qPCR, for which of course you need a target organism to be doing the qPCR for. But that would reduce the cost and increase the speed of the measurements we could take out in the field, so we could be a more effective regulator, because our compliance monitoring, our check of the performance of the regulated processes could be done more efficiently, more cheaply, possibly more of it and more accurately. So that's one big thing on my shopping list. Then it's to identify those targets that we need for regulatory monitoring, and we need the rapid molecular techniques to be able to exploit those. So I don't know if Rob Ferguson is on the call from University of Essex, but he's done some great work on using LAMP, a very portable form of effectively PCR, to identify individual organisms. But again, with this caveat that we have to know we're looking at the right organisms. So the wider shopping list is really understanding that background. It's making sure that we have the tools in place to routinely do that. And as Fred has said, we need to be able to do that consistently and intercomparably, so that I want my measurements to be able to be compared readily, perhaps with the Portuguese measurements, say. It would be really nice if we were doing this to a standard method. We need the agreed sort of sampling method, specifically geared up for DNA. So we're perhaps talking about cyclone samplers rather than the filters that we tend to use at the moment. We're talking about the bioinformatic tools. We need to be able to scan that set of information rapidly to identify what organisms are there. Okay, that's the start of the ask. Where it gets even more complicated is a big concern for us now is antimicrobial resistance. And we heard previously about the problem of azole resistance, which is something that's recognized as a problem here in the UK as well. And there's been some good research work, citizen science driven actually, trying to map out azole resistance in the UK, which is looking promising. There's also evidence that some of the processes we regulate, for example, composting sites, might be environmental reservoirs for antimicrobial resistant organisms. So is there something we can do with our processes to reduce that risk of antimicrobial resistance getting into the environment and hence getting into the exposure pathway for people? So we are beginning work on antimicrobial resistance studies, but early days yet. Again, we're going to need to identify the right markers for antimicrobial resistance, so we know what to look for in our sort of genomic database as we build it up. I think that's all I really need to say. In summary, then, we have the need for specific scientific research, for want of a better phrase, to characterize the atmospheric microbiome and to give us the target organisms that we can watch more specifically. We can use those in turn to inform our monitoring by targeting the right organisms and using more rapid measures, more perhaps indicative measures than culturing currently allows us to do. We can perhaps set up a network of surveillance monitoring to see how the background is changing. It would be rather nice, although this is very much my sort of fantasy perhaps, of having a national surveillance system for specific bioaerosols, so that we can track when organisms are appearing, how they're moving through the country, how perhaps resistance is developing and moving across the country. I think it's only by doing that kind of thing that we're truly going to understand both the causes and the consequences of antimicrobial resistance from these sources. Oza here would never forgive me if I didn't mention the non-biological measures of monitoring, and he's done some fantastic work with light scattering instruments and fluorescence instruments. Those have looked very, very promising in terms of identifying bioaerosol versus particulate matter. We've tried in the past to say, well, is particulate matter itself an appropriate surrogate for bioaerosol? Because it's much, much easier to measure. You can measure it in real time. You can see what each individual activity on part of the site is doing to the bioaerosol concentration. So we think there's great prospect there. At the moment, the instruments, of course, are big, expensive specialist instruments, and short of clonings are here. There's really no way of getting these in a lot of locations for routine bioaerosol monitoring. But if they can be used to identify perhaps windows of fluorescence, windows of particle shape, size, and fluorescent characteristics, that does offer the prospect of much lower cost tools, which might then be taken out in the field, even left out in the field, in the same way we do with other industries. We routinely monitor particulate matter or nitrogen dioxide or sulfur dioxide. So that would be a very useful thing to add to the armory at some point. That way you could pick up very rapidly when a problem was emerging and immediately go in and try and remedy that problem. I should say a quick word on mitigations. What a bit of a sorry story really for us was that when we were first starting to regulate bioaerosols, a lot of people were saying, well, we'll just put a biofilter on the end of our process. So an in-vessel composting site, churning up lots of bioaerosols, draw the air out, pass it through a biofilter, basically a huge box of sawdust or some other organic substrate. Now, these are primarily designed to reduce odor. Bacteria grow on the substrate, they break down the odor compounds, etc. So we did quite a bit of work looking at the performance of biofilters as a mitigation, and the results were quite an eye-opener, really, that in some circumstances, the sheer filtration physical barrier can help reduce bioaerosols. But it can also be bioaerosols. And if you actually get a low level of bioaerosol being produced from the source, then the filter itself can become a net source of bioaerosol. So it's actually making the process contribution worse rather than better. So we're very cautious about mitigation measures that people propose. And yet that is part of our work in assessing a permit. We have to be able to say, does this mitigation measure reduce bioaerosols? If so, which ones and by how much? We do that for a lot of industrial pollutants, and we need to find the right protocols, the right way of doing that for mitigation measures for bioaerosol. So yes, this time I think that is it. We need to know what are the organisms causing harm, or the parts of organism, in the case of, I don't know, endotoxin or plant fragments that cause respiratory irritation, etc. We need to use those both to understand background and to understand how best to do compliance monitoring. And we need to be able to keep an eye on what that background is, spread of antimicrobial resistance. Importing new pathogens is something else becoming a problem. And that's all. If you can solve that for me, we'll all be very happy. So thank you very much.
Thank you, Rob. Graham can sort out already some of your needs. It's nice to see in the chat. Thank you very much for this insight. And I think the two talks are great because they are effectively tackling back what do we need for the sampling and analysis, methodology harmonization, and ensuring we are able to provide a spatial but also temporal resolution. We do a lot on the snapshot resolution, but how we ensure we are providing the spatial temporal resolution. And there is also what the role of these emerging technologies. So you mentioned effectively molecular tools are now portable and the opportunity to bring them on the fields. But what really the role and how they can help us to investigate the microbiome and also the indoor-outdoor interface and how that will regulate, help to support regulatory needs, because we are detecting pathogens, or we're having quite a lot of high-level characterization. But what do we need to look for? What sort of pathogen we need to quantify? And